Okay, folks, today's talk is about cell membranes, a very busy place in the cell environment. So, clearly, we have the cell membrane as the outer edge of the cell's contents. And remember that it's not a two-dimensional circle, instead, it's three-dimensional sphere most of the time, or a sphere-like shape. Okay. Outside the cell has its own unique environment versus inside of the cell has its own unique environment. Now, both of them happen to have a lot of the same things in common, which is an aqueous environment, one that has a lot of water, but there's a lot of ions and other molecules floating around outside the cell as well as inside the cell, and those concentrations may be different from each other. But overall, we can say that this plasma membrane is a boundary that separates the living cell from its surroundings on the outside. Now, one special trait, if not the most important trait of the cell membrane, is selective permeability. And what that means, it allows some substance to cross it more easily than others. In fact, some things might be allowed fairly freely to flow through the cell membrane, and other things might be stopped entirely, and some things might be somewhere in between. Okay, so an example of selective permeability is, I'd like you to imagine if this yellow wall right here with these holes in it was ultimately a barrier that would let some things through and other things wouldn't be allowed through. Now, if you can look on the left-hand side, you can see some things here that aren't present on this side. And I want you to imagine the flow is this way from the left to right. Knowing the way this flows will allow you to determine what is let through and what isn't. For example, I see purple, yellow, and green molecules on the right-hand side. But on the left-hand side, I also see these big blue ones here and these big red discs, okay? Now, clearly these holes here are not large enough to allow those to pass through. And at the same time, the smaller molecules are able to fit through. That's an example of selective permeability based on size, okay? Some things are too big to flow through. Other things are small enough to get through and have no problem getting through. Now, this is actually what takes place in the human kidney. And these red discs, these are your red blood cells. And clearly, if the kidney is a filtering apparatus, and that filtering apparatus filters the blood, and what we want to do is we want to clear the blood of stuff that shouldn't be there and send it on its way in urine. So we're ultimately reforming urine from the stuff your body no longer needs anymore. But as your blood vessels are filtered, you'll notice those red blood cells don't go past it. Because if you had blood in your urine, you would know something's not right. We shouldn't have blood in our urine. So ultimately, it's a filtering mechanism, letting little tiny particulates, little tiny molecules get through that we don't need anymore, and keeping the stuff inside the blood vessel that we still need, like those red blood cells. As we mentioned earlier in our chapter, cell membranes are a mixture of lipids and proteins. And we said that the biggest part was made of phospholipids, which we learned during biomolecules, the most abundant lipid in the plasma membrane. Now, they get a special name to describe them, and here it is. It's a two-part word, amphipathic, okay? So amphipathic basically means that these molecules have a dual nature, okay, one or the other. And they contain regions that interact freely with water and regions that repel water, okay? So we have two different aspects to the molecules that build up the membranes. And let's review what those look like. So here we have a phospholipid head and we have fatty acid tails, okay? And what we learned was that the head up here was okay to mingle with water. So here's a watery environment. We'll say that's outside the cell. And here's a watery environment. We'll say it's inside the cell, okay? Now, these fatty acid tails, we said that they were made of mostly carbon and hydrogens over and over and over. And the nature of carbon and hydrogen bonds was that of being nonpolar. they share electrons easily, and they don't freely mix with things that are polar like water. Okay, water is our most famous polar molecule. And the head of a phospholipid has a lot of properties that allow it to mix with polar water. So the head is polar as well as what it mixes with. Same thing down here. This polar head of the phospholipid has no problem being in the water. But these fatty acid tails here, remember they were fatty acids, I'll put Fa for those, they did not mix with water. They were nonpolar, and polar and nonpolar things don't mix. So essentially what we have here is we have a barrier between two liquid compartments, the compartment on the outside and the compartment on the inside. Overall, we call the cell membrane a fluid mosaic model. And a fluid mosaic model states that a membrane is a fluid structure with a mosaic of various proteins embedded within it. Okay, so in the world of art, a mosaic would look something like this. Okay, you can see there's a lot of these pieces of broken glass that are surrounded by some kind of substrate. Maybe it's cement, maybe it's something else. But these pieces of glass in here that are trapped in this matrix of whitish gray, they're very much like the proteins that we have inside of our fluid mosaic model. So we have a sea of lipids with proteins flowing through it. So all these blue and purple things here would represent those proteins, and all this white area here would represent the lipids. Now, in reality, does it look like this piece of art with glass and perhaps concrete or something? No. Instead, there's a lot more lipids than there are proteins, which we'll see in the next slide. Oh, sorry, it's here on our slide. All right, so all these red parts, these are those heads that are equivalent to this and this, and then these greens down here going into the interior, those are our fatty acid tails. So you can see how many lipids we have here, and then these blobs that are interspersed throughout, those are your proteins floating in that sea of phospholipids. Next, taking a closer look at our phospholipid bilayer and actually cutting in from the side to take a look at it. Here you can see our proteins colored purple, big purple blobs. We say that's typically how they're represented in textbooks. And these proteins are spanning the membrane all the way from the top to the bottom in some cases. In some cases not. Like for example, you see this little chunk down here that doesn't make it into the membrane at all. They have different names and they have different functions. But let's talk about the proteins spanning the membrane right now a little bit. And the key term I want to use here is hydrophobic for the region that is inside of the membrane here. So again, proteins are made of amino acids, and a lot of them, right? So this area right here that's within the fatty acid tail region of this cell membrane would likely have very similar properties to those fatty acids, and we said they're nonpolar. So what we could say and what we could deduce is that we have nonpolar amino acids, I'll just put AA for that, in this region here. So whatever amino acids are making up this protein, they're also going to be the nonpolar because they're mixing here with these nonpolar fatty acid tails. Now, meanwhile, on the ends, the ones that are exposed to water on both sides, so like this right here and this down here, these amino acids must be of the polar type, okay? Because they're sticking out into the watery environment of the extracellular fluid, the area outside the cell here. And this part here, I'll just call it polar. I could probably fit it in there. Polar AAs here too. Notice how it is also in a watery environment. This is the cytoplasm. And those amino acids must be free to mingle with an aqueous environment as well. So we have this nonpolar water disliking region in the center here. And then we have amino acids that don't mind water because they are polar themselves. And we use the term hydrophobic, hydro meaning water, phobia means fear, okay? So we have ultimately the amino acids that fear water in the middle. Philic means a like for something, an affinity for something. So for those, you're going to have amino acids up here that have an affinity for water, hydrophilic. Another important molecule that exists within the cell membrane is the steroid called cholesterol, part of the lipid family. It's going to embed itself every so often amongst those phospholipids because it has a job to do in order to keep our cell membranes fluid. Okay, so cells are not rigid at all. They're not like a rock on the outside. There's a lot of flexibility to them, and temperatures will affect that flexibility quite significantly. For example, let's imagine that we have some warm temperatures that a cell exists in. We'll say 37 degrees Celsius, which is just about normal human body temp. Okay, that produces a bit of vibration, a bit of motion for molecules. The warmer it gets, the more they vibrate, until one point they start flowing over each other. And cholesterol, which is this molecule right here, is ultimately going to anchor this in place and not let it get so fluid that it will start coming apart. So we could say cholesterol restrains the movement of the phospholipids. It kind of has little anchoring positions in here just to keep it solid, okay? Because we know temperatures can go above 37, like when we're sick and we have a fever, and we wouldn't want our cells literally falling apart. So cholesterol will kind of anchor them in and keep those cells together. On the flip side of that, there's plenty of organisms that exist in the cold, and we don't want their cell membranes getting too rigid. So another thing that they do is they provide little spacers so all of these nonpolar legs of the phospholipids don't get all jammed up together with tight packing and cause this to solidify. So ultimately, the cholesterol serves as a buffer in both directions. Doesn't let it get too fluid, but also doesn't let it get too solid upon, exhibiting cold temperatures. For example, like we'd have in the fall and winter of given areas outside, and things have to survive through that. So the question at the bottom, which molecules are cholesterol? It's going to be these. Sometimes people will refer to them as 3 hexagons in a doghouse. So if you look at them closely, you can see 1, 2, 3 hexagons here, and then you see a pentagon. So these 4 geometric structures, 3 hexagons and 1 pentagon, or 3 hexagons in a doghouse, is the chemical shorthand description of a steroid molecule like cholesterol. Now, on the outside of the cell, we have molecules that stick out that their job is to ultimately provide recognition sites for other cells, cell to cell or system to cell, and I'll get into that in just a minute. But these surface molecules that stick out are often carbohydrates, and you can see them here in these chains sticking off the surface. Now, when a carbohydrate sticks off of a lipid, like we see here, this is called a glycolipid now, okay? The glyco is the sugar part, and then the lipid, in this case, would be our phospholipid right there, okay? When we have a carbohydrate bonding to a protein, it's called a glycoprotein. Again, here's the glyco part, the sugar, and here's the protein part down below. Now, again, we said that this was a recognition molecule, okay? So I want you to think of these chains sticking off as sometimes the cell has to reinforce and remind the organism that it sits in that, hey, I'm a part of you. So this sign might be tailored directly as, like, imagine it's one of your cells. It could be your name in chemical language that says, hey, immune system, don't attack me. I'm part of you. I'm part of this body. And another cell might have the same thing coming off of it, and the two interact together, and they know they're part of the same tissue. But if another surface molecule was present inside the body and your immune system was checking up on it, and it wasn't this particular shape, your immune system might actually attack it, thinking it's some kind of foreign invader. Okay, this often happens when people get organ transplants. The cells that come in are not technically part of you, so they'll have a different chemical signature on these carbohydrates than your cells do. And your body would look at it as a foreign invader and most likely try to attack it. So how do we prevent that from happening? How do we have successful organ transplants? Well, for that, we will typically give the patient something called immunosuppressive drugs, which will basically calm their immune system down and let it tolerate more than it normally would. So if someone got a new lung or a new liver, some new body part, they could actually tone down the volume on their immune system response, and then slowly over time, it kind of gets used to it. Now, typically, a transplant won't last forever. Eventually, your immune system is what it is, and it will recognize, hey, there's something not right with this. And organ transplants may not always be forever. Your immune system may catch on and eventually start damaging that organ that has been transplanted. But there's plenty of successful stories where people can have organs from someone else over 10 or 15 years even. There's also a sidedness to membranes. So I want you to look here at this would be the top, or the we'll call it the extracellular fluid on this side, and on this side down here is our cytoplasm. So what that tells us is that we have a sidedness to the membranes that aren't the same. Look at all of these things here on the top, and you can see you don't see a mirror image down here on the bottom. So there's definitely not a mirror image of things on either side of the membranes. Instead, they're very, very different, okay? These gray circles would represent those carbohydrates we were just talking about. These here would be proteins sticking out. And on the other side, you can see we have these twisted, red and pink balls here, and these are part of the cytoskeleton we've talked about before. They're part of the machinery that allows a cell to essentially put out a pseudopod and wrap around something, outside of, or move the cell. So what we can say is there's an asymmetrical distribution of proteins, lipids, and carbohydrates in the plasma membrane. Okay, asymmetrical means not a mirror image. The two sides are different. Now, as far as building this elaborate cell membrane structure here, we have to go back and reflect on those organelles from inside the cell and remember who helped build it. So we said it was part of the endomembrane system that eventually would send products from the very middle of the cell near the nucleus outward to the outside. And the first stop, if you will, past the nuclear membrane is the ER, the endoplasmic reticulum. It's there where proteins are made on those ribosomes that are studded to it. It's there where lipids are made, which will be passed on further towards the outside of the cell. After the ER, whatever was built there takes a ride to the next big structure, which was the Golgi apparatus. And it was at the Golgi apparatus where we saw things get wrapped up in nice vesicles. Perhaps a sugar was added or transferred or removed. So it's kind of a sorting and repackaging area. And eventually from here, we have vesicles once again take those products to likely the plasma membrane on the outside of the cell, and then perhaps become part of the cell membrane's unique set of structures that you see here in this picture. All of those unique structures are going to aid in its ability to be selectively permeable, okay? Now, there's no doubt about it, cells must exchange materials with their surroundings. They're just like you and me. The fact that we have to obviously take in nutrients and we have to get rid of waste. Cells have to do the same thing, and that's controlled by the plasma membrane. They regulate the cell's molecular traffic. Now, let's take a look at some things that have a green light to get in the cell with very little problem. Oxygen, carbon dioxide, small nonpolar molecules, okay? They have the ability to go right through, okay? An oxygen atom is very small, very nonpolar. Same thing with carbon dioxide, okay? Carbon dioxide leaves cells as a waste product. And it's got to have a quick entry and quick exit to really meet the needs of a cell in both intake and output. Now, the things that get yellow lights and red lights in that traffic are different. Glucose, large polar water-soluble molecules, ions, water molecules. These things get stopped. Let's recall glucose for a second. Remember that big hexagon? Okay, how is this going to get into this cell by slipping through the phospholipids? Not very easily, right? In comparison, the oxygen molecule is about like that in size compared to this big old glucose right here. Glucose is also loaded with hydroxyl groups sticking off it, very water-like, okay? That is a no-go for a nonpolar membrane, okay? So they get stopped. Other large polar water-soluble membranes, same thing. All these charged ions, they don't have an easy time getting through either because, again, polar molecules like water, they're charged, and this is a neutral, non-charged area in here, and you just can't pass through it very freely. So if you're charged or you're polar or you're big, you are not getting through like something small and simple like carbon dioxide and oxygen. So how do these things get in then, these larger things? Well, they're going to make use of what's called a transport protein. So those big protein blobs that we said were embedded in the membrane that allowed some things through and other things not to go through, they're going to be a big helper in that effort. They will allow passage of hydrophilic substances, of things that like water. So for example, these red molecules here are going to pass through this channel because otherwise they couldn't get in. Okay, this carrier right here is going to catch these blue ones, make a little turn within the membrane, and then dump them on the inside. Okay, so this is the help that those molecules need to get in. And meanwhile, we said those small little ones, they could be the ones that pass right through. So why can't they get it on their own? So we're all over it now because they're charged the wrong way. They have charge and most of the membrane doesn't. They're too big and they're going to need some aid to get in. Channel proteins allow that small tunnel to provide the place for polar substances to move through. So this is what we call an aquaporin, and this was discovered as a way that water could go in and out of cells because it couldn't go right through the membrane itself. It has to have a little tiny channel to get through because of the center of the membrane and the chemistry that it has. So those are called aquaporins, built specifically for chains of water to get through. And you can see they're very, very narrow. Sometimes, you know, just a few molecules at a time can go through like a chain. And we're going to learn why water would go into or out of a cell in a little while. Some other transport proteins are called carrier proteins, and they're going to bind to molecules and change shape to shuttle them across the membrane. These transport proteins are going to be very specific for the substances that they move. So for example, this one here has this little notch carved out for it, which is built specifically for this orange triangle or red triangle. Okay, clearly the blue one wouldn't fit in that site, again meaning that it's very specific. Now what about this yellow one? Couldn't it come in at an angle and fit in there? Perhaps, but perhaps this area here is lined with amino acids that happen to match with the ones right here, and that the ones that line this corner and this corner do not bind well with those same amino acids that line that little active site, that line that little receptor port, okay? So they're very specific for what they let in. Now, the movement across the membrane is thought about in two different ways. One, where the movement is passive and happens on its own without an energy expenditure. And two, active, meaning that you have to pump energy into the process in order to move substances across the membrane. So we're going to start with passive transport, and that's the one where there is no energy investment. So passive transport means it just kind of happens on its own because of the concentration of things within a cell. So we'll start with the idea of diffusion, and we've been here before. We've talked about diffusion, and it was the tendency for molecules to spread out evenly into the available space from an area of higher concentration to an area of lower concentration. Okay, so if something's heavily concentrated in one area, it's going to move to those other areas over time. It doesn't take any work for that to happen. It just is due to random collisions of particles flying through the air or floating through a liquid, banging into each other, banging into walls, and eventually making their way across an area. Now, In the beginning, when that first starts, there's typically what we call a net movement in one direction, okay? And that will continue until a certain point, which we call equilibrium or a dynamic equilibrium, where as many molecules will cross one way as that go the other way in the other direction, okay? So it's kind of a place where it finally has become equal. Here was an example we looked at once before. So let's imagine we release a whole bunch of yellow dye in a sample of water that has a membrane splitting a left chamber and a right chamber. In the beginning, you're going to have a net diffusion of these molecules to the right as they try to, as they will reach open areas that they weren't in before. And that net movement will continue, that net diffusion, as long as there's more on one side than the other. But eventually we'll reach a time in which we'll get to an equilibrium where we have roughly the same amount on one side as the other, and the rates of going forward and backward are essentially the same. Now down in the bottom one, what if we had like a yellow dye and a purple dye that were released at the same time? Eventually each one would spread due to its own net diffusion, purple to the left, yellow to the right, and that would continue again until at one point there's an equilibrium. So like here we have 1, 2, 3 purple, 1, 2, 3 purple. Here we have 1, 2, 3, 4, 5, 6 yellow, 1, 2, 3, 4, 5, 6 yellow. This would be the equilibrium point. Now, could you have, some travel to the other side and be uneven for a moment? Absolutely. But overall, the net is going to be the same. So it will probably even out within moments again. So again, with diffusion, substances diffuse down what we call the concentration gradients, which was the difference in concentration of a substance from one area to another. So if this is a cell right here, clearly there's more red substances in this one. And those molecules, just due to random nature, are going to be finding their way out to eventually reach an equilibrium with the outside. This one over here is the reverse of that. There's a lot more particles on the outside than the inside. So a chemical driving force, which is ultimately diffusion, is going to eventually bring those red particles in until they reach an equilibrium point as well. No work is going to need to be done for passive transport to occur. And again, there's no energy required to make this happen. It happens on its own. Let's talk for a moment about the effects of solute concentration on water balance. Now let's review solute. Solute is just dissolved stuff, okay? And these little brackets mean how much dissolved stuff there is in water, okay? That will have an effect on where the water is and whether it's going to go from one place to another, okay? So let's start with this, osmosis, okay? Osmosis is the term that we use that describes water diffusing across a semi-permeable membrane. Now, the difference here and what we were talking about before is that this is water moving, okay? This isn't little red dots. This isn't little green dots. This is straight up water traveling across membranes. So water will do that, and it will travel across the membrane from a region of low solute concentration to a region of high solute concentration. All right, let's break that down. All right, if something has a low solute concentration, it means there's not a whole lot of stuff dissolved in the water. So what that means is that there's a lot of water there, okay? So low solute is equal to high water, okay? Now, high solute concentration means there is a lot of stuff dissolved in the water. So high concentration equals less water, okay? Now, water is going to diffuse from the low region to the high region, okay? So what that means is water is going to kind of move in the direction where there's more stuff. Water is actually referred to as a great diluter, okay? A dilute solution is one that there's a lot of water in. Like if you made a powdered lemonade or Kool-Aid, and it was mostly water, you'd taste it and go, this is kind of tasteless. This needs more Kool-Aid powder. This needs more, iced tea powder, lemonade powder, whatever it is you're making. Versus if something has a high solute concentration, you put too much sugar, too much drink mix in there, and you taste it, and you're like, oh, wow, this needs water because it is really strong. So water, you'd put water in there to dilute it to make it less strong. Water on its own will travel into regions like that. So if you look at this picture down here, you can see there's these green particles, whatever substance we want to make those, and then they have water molecules around them. And on the left side, you can see there's a lot more water than there are solute particles. On the right side, you have a lot more green solute particles than you have water. So this is a lot more highly concentrated. So the question is, which way will the water go? And if we read this statement again, water diffuses across the membrane from the region of low solute concentration to the region of high solute concentration. Well, this is the high solute concentration over here. This is the low. Water is going to go from low to high to try to dilute it. So ultimately, what we should see is a left to right motion of water in this case. And putting it into motion, you can clearly see we have water mainly going from left to right to dilute what we have over here. Okay. Now, there are a few that water molecules that go back the other way, and that's just what happens during the overall process. But net, in terms of what's happening, we definitely see a right to left type of motion as that water tries to dilute what's on the right-hand side of that membrane. That's osmosis. Here's another slowed down version of osmosis. This is like the original U tube right here. It's literally a tube and a U. And what we can say about it is that in this U tube right here, we have equal volumes of water on the left and the right. There's a semi-permeable membrane down the middle. On the left-hand side, you got 1, 2, 3, 4, 5, 6 green dots, green molecules. On the right, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, right? So you have a much more heavily concentrated solution over here. So what happens, okay? And we'll say that those green dots are sugar, by the way, over here. Make sure we know it's sugar. The green molecules are too big to fit through these little gaps. If we look at the little cutout down here, look at these big green molecules. There's no way they're fitting through this span right here. But you know what can fit through that span? Water molecules, and they are free to move. So the question is, which way will water go if this side over here is a lot more concentrated than this side? Well, look no further than the picture on the right. This is one little few moments later. The water has moved, and it has gone in this direction over here through these gaps in the semi-permeable membrane. And it did that because it was chasing where the particles were at. It was working to dilute these particles over here and form a new equilibrium. And in doing so, look at the volume. The volume actually decreased on one side. So this thing literally defied gravity. Yeah, it did. The water moved down through here, went up in this tube here due to the chemical interactions between water and these molecules. And this U tube will permanently stay like this as long as nothing else happens. Okay, so we clearly have a net movement of water from the left to the right as water tries to dilute this side over here of the U tube. Once it's like this, now you have the same concentration of sugar. You're at equilibrium. Now, we're going to stay on the topic of water movement here, but we're going to bring in some of the structure of the cell when we talk about it. Some cells have cell walls, other cells do not. So that puts a variety of scenarios in play as we talk about water movement in various kinds of cells. So let's do a vocab drop here of the word tonicity. Okay, tonicity is the ability of a solution to cause a cell to gain or lose water. Okay, so imagine any type of cell that's in your mind, animal cell, plant cell, fungal cell, whatever you want. There will be movement of solution, movement of water in and out of cells of every organism type. Okay, now what we're going to look at is we're going to look at a few labels of these solutions, which is going to help us determine what happens with water. First, isotonic solution. Okay, so iso roughly means equal. Okay, an isotonic solution is when the solute concentration is the same as that inside the cell. no net water movement across the plasma membrane. So things are fairly equal on both sides. That's isotonic. Next, we have hypertonic, okay? Now, hyper, in terms of scientific prefixes, basically means above or higher, okay? And what that means is that the solute concentration is greater than that inside the cell. So what's outside the cell in the solution is higher in concentration. And when this happens, the cell will lose water, okay? So imagine we have a cell, and we'll just say the area around it out here will have high concentrations of, let's say, salt, okay? Inside the cell, you have low concentrations of salt. Now, because water flows into areas that have high concentrations, water is basically going to be leaving the cell, trying to dilute what's outside of it, okay? So you're going to have a lot of water leaving, and your cell is going to lose water and volume by doing that. The other scenario is what's called hypotonic, and hypo means lower or below in biological prefixes. So the solute concentration is less than what you find inside the cell. In that scenario, the cell would gain water. So let's say here's the area outside the cell. Here's the area inside the cell. Let's say we have high salt inside and low salt in the fluid outside, okay? Because this is high salt concentration, water is going to leave that exterior area and start coming into the cell, making the cell gain water and swell in size. Now, let's look at the difference between animal cell and a plant cell in various, we'll call it tonic solutions of different tonicity. Okay, let's imagine this blue color here represents how much stuff is dissolved in the solution around a cell. Light blue, not a lot. Medium blue, a medium amount. And then deeper blue or dark blue here, we have a lot of stuff dissolved in the area outside the cell. Okay, now let's run through for an animal cell versus a plant cell. When a red blood cell, and that's what this is, by the way, is put in something that is mostly water and not a whole lot of solute dissolved in it, the cell becomes saltier than the area around it. Water comes into the cell and lyses it, which means it basically explodes this cell. There's no cell wall to hold it together, and the membrane just essentially explodes. Okay, can't hold any more water. Now, if there's a little bit of salt outside of it, notice that this is a pretty good looking red blood cell. Okay, it has this nice oval shape. Officially, it's called a biconcave disc, but you can tell it looks really good, really healthy. And that's because a little bit of salt is okay for a red blood cell because it also has a little bit of salt inside of it. And us as human beings, you know, we're big bags of salty water. And having this type of cell that enjoys that environment where it's at equilibrium with it is perfect. Then imagine that our cells are surrounded by a really salty fluid, lots of stuff around it. The water leaves our red blood cells, and what happens? They shrivel up, okay? Because they're losing all their water to the outside. Not good for a red blood cell in this case or this initial case. But then in a plant cell, we see something a little bit different. Here, when there is very little solute in the water, mostly just water, and water starts coming in, that's actually really good for a cell, okay? Because it makes the plant turgid or normal, okay? The cell is kind of like busting at the seams, but it's not going to explode because it has a cell wall around it like all plant cells do, okay? Now, as you start adding stuff into the water, like salt, for example, notice that this plant cell here is not busting at the seams like this one over here. Instead, it's just kind of square, and it leads to a flaccid state where, you know, overall, if all the cells in the plant look like that, it's just kind of droopy, not really that healthy looking, not vibrant. And then if I put a lot of salt in water and I give that to the plant, basically all the water is going to start leaving the cell itself, going into the exterior environment and pulling away from the cell wall, plasmalizing each of the plant cells, and eventually killing the whole plant. Okay, so that's why we don't put salty water on plants, because ultimately it's going to do this to the plant. First it'll go flaccid, then it will plasmalize, and all the cells will die because there's not enough water to keep it alive. Okay, what we want is pure water on the plant, so it is, full of water in this turgid state, which is normal. So what we can say is that osmoregulation, this is a new term for us. This is the control of water balance. It's one of those necessary adaptations for organisms. And we said that one of the things that living cells do is they adapt to their environments. And this is an adaptation that they have. So they have just the right amount of liquid in them, and they get to their balance, their optimal balance, by either purging water or allowing water to come into them. Going back to talking about proteins helping things in, there's a name for that, and we call that facilitated diffusion, because on its own, this orange molecule here might have a hard time getting through the membrane, but if there's a protein that's built specifically for it, it can help it get across. Okay, so in this case here, you see this little purple flap on the membrane protein. It goes up to accept the molecule in. Once it's in, there's a little chemical reaction that folds it down. It kind of guides it, facilitates it through the protein into the interior of the cell, and then it pops back open here, ready to accept another one. When it catches it, it will again push down, allowing that to go from one side to another. So that's facilitated diffusion. It's aided by movement, aided by proteins. We also mentioned earlier the channel proteins that basically give the corridor that allows things to float through, like the aquaporins. Carrier proteins, a little bit different shape here, might be able to completely change their shape as things float into this holding spot right here. The whole protein might change shape, pivoting its point right here, allowing it to dump in as well. So solutes have a couple different methods for getting in channels and carriers. We said that water, aquaporins, would kind of take this type of route into a cell. And there's even other ones that exist within our bodies. They're called ion channels that specifically help those charged particles get in. And they often are gates. So, gates can pop open and close in response to a stimulus. So your nervous system cells work like this. A stimulus might cause a gate to pop open. Like if I have let's say this is my cell membrane here, and these are my proteins that are, here's the outside, here's the inside. There might be a gate right here, which, given a proper stimulus, might pop open, allowing a whole bunch of stuff to flood into the cell when it's open. And then that gate will slam shut at a given moment, no longer allowing those ions to flow through in this direction. Okay, that would be an example of a gated channel. And when we study the nervous system, we'll talk about those. So again, big picture is when you have more of one thing on one side of the membrane than the other, diffusion tells us that it's going to try to get into the cell where there's less of it. And it seems like getting all this stuff in would require a whole bunch of energy, but remember that facilitated diffusion is still going to be, qualify as passive, because those solutes are moving down their concentration gradient. They just happen to be controlled a little more by these gatekeepers within the membrane itself. Now, some transport will require energy, okay? So that is going to be called active transport. And this is when substances are going to move against their concentration gradients, okay? So imagine you're standing in a river and you're trying to push the water back from the direction where it came. It would take a lot of energy to do that, right? Well, that's the same scenario we have at the cell border. And sometimes we try to jam things like these positive charges here. You can see them. In this case, that red arrow means they're trying to get in this area where there's already a high amount of positive charges. This is going to take energy to make that happen, okay? Now, in biological organisms, that energy is going to come from that molecule called ATP. ATP was a nucleic acid. It was the energy currency of the body. And very specific proteins are going to help with that process of pushing things into cells or out of cells where there's already a high concentration on the other side. So let's check for a second and see where we are with our types of transport. So over here on the left, we have diffusion, simple diffusion, small nonpolar molecules just gliding right through that phospholipid bilayer. Next to it here, we can see there's a channel protein, and next to this one here, we have a carrier protein, both of which are passive. They're still passive, high concentration to low concentration, except these need facilitators. Maybe they're just too big and clunky to pass through the cell membrane itself. And then over here, we have the example of active transport. And you can see on the outside of the cell, you got a couple squares. And the inside of the cell, you got 1, 2, 3, 4, 5. And you can see these squares are being pushed in with the help of ATP. Okay, so this is an example of active transport, going from low concentration to high concentration. Last thing we want to talk about is bulk transport. So bulk transport means really, really big stuff in and out, okay? So we just saw the mechanisms for getting small things in and out. But what about really big stuff? Polysaccharides, proteins, things like that. Well, for that, we're going to need energy to get it in, and vesicles are going to be involved as well. Okay, so let's start with what's called exocytosis. That's moving stuff out of a cell. And let's imagine all these little red molecules gotta go. Okay, now they are currently enclosed in a membranous vesicle. And this membranous vesicle is going to basically fuse, merge with the cell membrane, which membrane is membrane, so it's made of the same stuff. So when it does, you can see that the contents on the inside slowly get dumped to the outside, and that vesicle now becomes part of the actual membrane itself. That's what we call exocytosis. Exo means exit. Okay? Secretory cells use this process to export their products. A secretory cell is something that secretes stuff. Okay, and you have a lot of cells inside your body that secrete things into, say, your bloodstream, for example. So hormones is one of the things that gets secreted into your bloodstream, and then the hormone it'll travel through your bloodstream until it reaches a target cell to do something. Okay, so this is a very common process to get rid of waste and or secretions that your body may need. Okay, here's another look of it. Molecules, they're currently trapped in a lipid bilayer vesicle. This vesicle slowly starts merging with the cell membrane and starts dumping the contents outside. That's exocytosis. Next, we have endocytosis, which is the reverse of exocytosis. You're going to take in large things from the outside by forming vesicles from the plasma membrane itself. Okay, and we'll break that down into 3 separate versions of it: phagocytosis, which is cellular eating; pinocytosis, cellular drinking; and then something called receptor-mediated endocytosis. Okay, so first we have phagocytosis, is where a cell engulfs a particle in a vacuole. All right, so let's look at the picture here. We're going to imagine this here is our food. Okay, this food is something the cell wants, so it's going to extend out portions of its cell membrane. We call those pseudopods, and that was basically formed, it's helped out by the cytoskeleton. They blob around your food source, and one moment later it's now wrapped in membrane on the inside of the cell here. Okay, and we said the next likely thing to happen was that vacuole would fuse with a lysosome. So a lysosome with its digestive enzymes will then fuse with this. The enzymes will essentially get in here and start breaking down this food into really small pieces. The food vacuole will come apart, and then we can use all those little spare pieces for other things inside the cell. Here is an example of an amoeba. An amoeba is a little tiny single-celled organism blobbing around a bacterium. This whole thing right here, this is a bacterium. This is its last day. This is its last moment, because that amoeba is going to keep going with this and this until it's 100% engulfed that bacterium around it, and that will start digesting it on the inside. So good day for the amoeba, bad day for the bacterium. Next, sometimes cells need to take a drink. Pinocytosis is the same thing as that phagocytosis, except it's just allowing a blob of fluid from the outside and whatever happens to be in that fluid to come on in the cell. So now you have a vesicle that was made from the membrane itself that has extracellular fluid in it from the outside, as well as whatever these molecules are here as well. And here you can see a whole bunch of those coming into the cell lining in a blood vessel. Okay, so taking a little drink. The last type I'll just talk about real quick is called receptor mediated. And if I was to draw a cell membrane, you might have some really special receptors sitting in a pit that is on the outside of the cell. And to trigger a receptor mediated event, what we'd have to have is you'd have to have some molecules that would fit in these receptors just perfectly. Okay, when they do, that would be like the little chemical trigger that tells this membrane here to pinch off in this direction, bringing this in. And again, you might need those molecules from the outside, but now basically you have on the interior of the cell, you have these little tiny receptors on the inside of this vesicle, which was just on the outside of the cell a moment ago. But these little tiny things is what that cell needed, so it brought it in using receptor mediation. And that's all that one means. Okay, so it's a little chemical trigger that causes it to come on into the cell. All right, so this concludes our talk on cell membranes, a key part, very busy interface of every cell, and absolutely required to understand the metabolism of cells and how nutrients get in, waste gets out, and how that border operates. So I hope you learned a lot, and we'll see you at the next lecture. Bye-bye.